Yeah, welcome everybody to the first trainee seminar of the fall session. We've got two really exciting talks today. I'm looking forward to hearing about both of them. So the first is going to be from Zachary Barrand. He is a PhD student in my group at Northern Arizona University and also at the Translational Genomics Research Institute. Zach graduated with a undergrad degree in biological sciences at NAU, where the focus was on co-infections with tick-borne disease. And then now he's focused more on the mosquito side of things, looking at Culex population genetics, as well as West Nile virus spread over time and space. In the second talk, which I wish that this program was going on here in Northern Arizona, but it's focused on training STEM students in mosquito surveillance and identification. And that's going to be given by Abdoulaye Zerbo and Mayra Espinoza. And if I did mess up your name, please feel free to reintroduce yourself at the beginning of your talk. So Zach will have about 25 to 30 minutes to give the first presentation, and then we'll have the same amount of time for the second presentation. And so with that, Zach, do you think you can share your screen?
Yes, I can share my screen. Okay. Can everyone see my screen?
Yes.
Yes.
Perfect. Okay, so the CDC has reported over 56,000 cases of West Nile virus since its initial introduction to the United States in 1999, with the Southwest reporting human infections and non-human activity annually, making the virus and its vector of particular public health importance in the region. And so I'm just going to first introduce West Nile virus. And so West Nile virus is a single-stranded RNA virus, and it is a neurotropic pathogen. So it's able to infect host nervous system tissue. And within humans, symptoms can range from asymptomatic in most cases to neuroinvasive disease such as encephalitis and meningitis, depending on host immune function. And West Nile virus is a zoonotic pathogen transmitted by mosquitoes. And so that means it's maintained in the natural environment between a reservoir host and a vector. And for West Nile virus, our reservoir hosts are going to be bird amplifier hosts, and our vector is going to be a mosquito, specifically mosquitoes of the Culex genus. And this West Nile virus is maintained because our amplifier bird hosts, when they become infected with West Nile virus after being bitten by an infected mosquito, they produce high enough viremia within their blood that when an uninfected mosquito bites them, that mosquito becomes infected and then can transmit it to an uninfected bird. And so it's in this natural cycle between bird and mosquito that West Nile virus is maintained. And mosquitoes can sometimes feed on other hosts, such as a horse or a human. These are known as dead-end hosts. And so these are dead-end hosts are hosts where when they get bitten and become infected, they don't produce high enough levels of viremia within their blood that they can infect another mosquito when it bites. And so they're dead-end, they don't contribute to the maintenance of West Nile virus. And when we look at the incidence of West Nile virus over space and time, or over time and throughout each U.S. county since its initial introduction in 1999, we can see in New York, we had human infections reported. And then by 2000, we saw it expand and we saw non-human activity with human infections. And by 2002, there was a big explosion of West Nile virus. And in 2003 was the first reported cases of West Nile virus within Arizona. And by 2004, it spread across the entire continental United States. And as we progress through the years, we can see that human and non-human activity was bouncing around by county every single year. However, in the Southwest, we can see that we have persistence of West Nile virus in those counties. In particular, if we zoom in on Maricopa County, the upside-down L county in Arizona there, we can see that there were human and non-human activity reported every single year. And as we come into closer to current time, where in 2020, we kind of saw a low year, but by 2021, it kind of expanded again. And I want to point out this year in particular for Maricopa County, because this was the largest year on record for any county in U.S. history. So Maricopa County had the highest number of cases in U.S. history in 2021. And we continue and progress into 2023, where we have our current data. And having this data is great tracking through time. However, is there a way to monitor West Nile virus before it even gets into the human population? And so we are able to use mosquito surveillance as a way to monitor West Nile virus. And so on this plot, we can see on our left y-axis, we have our positive or West Nile virus positive mosquito pools. And on our right y-axis, we have our count of human cases. And throughout the years, we can see that mosquitoes and humans are the number of positive mosquito pools and human cases align pretty closely, where West Nile virus positive mosquito pools explain 76% of the variation observed within the human cases. And so we can use mosquito surveillance as a way to monitor environmental load of West Nile virus and it can be used as a predicting tool for spillover into human populations. And so when we zoom into Maricopa County and look at the density of West Nile virus positive mosquitoes over time, where we don't have a heat map, those are going to be our negative traps. Where we have our heat map, we're going to have low density in blue and high density of West Nile virus positivity in yellow. And we can see year over year, while we still see some moving around of West Nile virus positivity in our mosquito population, we have identified hotspots of high density over time. And so we kind of have at the 101, 202 junction in the Phoenix metro area. That's going to be a hotspot that we've seen over time, as well as we have a couple over in the West Valley. And when we look at 2021 in particular, which was the largest year on record for human cases, it was also the largest year on record for positive mosquito pools. And so West Nile virus was everywhere in the county. However, what's significant is that our hotspots did not change for that year. And so we're still seeing the same hotspots that we did in previous years. And so this is where, what is our goal for our study? And so we want to be able to use mosquito surveillance as a tool to track West Nile virus over space and time throughout the region and identify hotspots of concern so that we can better inform our vector control partners in their mosquito control efforts. And so, like I said, we're working with our mosquito control partners, quite a few throughout the southwestern region of the US. And what they do is they go through and they trap all of the mosquitoes, typically weekly traps. They bring the traps back to their labs, sort the mosquitoes by trap and species, and pool them by 50 mosquitoes per pool and 5 pools per trap. Males are discarded as they don't contribute to the maintenance of West Nile virus, as they don't take blood meals. And then the resulting pools are tested for various arboviruses, including West Nile virus, via reverse transcription qPCR. And then that's when we come in. So we take the positive West Nile virus mosquito pools from our collaborators. We reverse transcribe the RNA virus into cDNA, and we perform a tiled amplicon PCR approach to amplify the complete genome of West Nile virus. We then use a custom universal tail approach and sequence that on a Illumina platform. We then take the resulting reads and we trim them and perform a reference-based assembly using the New York 99 genome, using BWA. We take our consensus sequences for each of our genomes and build a phylogenetic tree and to tell us the phylogenetic relationships present within the region using NextStrain. We then perform a transmission network analysis to understand our source locations and what traps are contributing the most to the networks and the particular populations that we've identified in our phylogenetic analysis. And so moving into our NextStrain, I have a QR code there at the top if you'd like to scan it and interact with our NextStrain further, but I'm just going to go and highlight over it. So our NextStrain contains over 900 genomes that we've contributed ourselves from our group, as well as over 300 publicly available genomes. And in this, following that analysis, if we look at the exact samples that we've contributed, we've identified a Maricopa County endemic clade with an introduction last known in 2011. And this we've also confirmed with an internal phylogenetic analysis where we've included an additional 1,000 genomes from the West Nile virus 4K project. And in that analysis, this Maricopa County endemic clade is still present. And so if we zoom in to a section of the West Nile virus endemic population or endemic clade, we can see here that we've had numerous introductions into other counties from Maricopa County. And so I'm going to point out here at the bottom, down in 2019, there was an introduction from Maricopa County into Clark County, Nevada, causing an outbreak of West Nile virus. And then I want to point out the one up here towards the top in 2020, where there was an introduction into Riverside County, California, that is now a population that is being maintained in that county. And going back to, looking exactly at the 2021 samples that we've sequenced, we have quite a handful of populations that pop out that emerged within 2021. And so we have a population here at the bottom, and then a couple others here in the middle. And we have one right here, this big one right in the middle, that's going to be an admixture population where there's movement between Maricopa County into Riverside County, California, and back into Maricopa County. And so we've identified 4 Maricopa County endemic populations. And so they've had exportation from Maricopa County, but no new introductions into that population. And when we zoom into the first population, we can see that this is a population that is centered around kind of the eastern part of the valley or eastern part of the county, around Tempe, Mesa, and Chandler. And when we take this population and we perform a network analysis, we performed a degree centrality analysis, which basically just tells us what traps are connected to each other. And so how important a location or host is within the transmission network, given the number of times that a disease emerges from or to that point. And so we can see on this plot here that our connections or the number of connections that we have, which is our x-axis, most of our traps only have one connection. So they're not contributing much to the network. Whereas we have a couple of traps that have a lot of connections. They're contributing more to our network. And then we also performed a source hub ratio analysis. This told us basically what are our source locations, what are our sink locations. And so where did it come from and what traps are not as significant in the maintenance. And so our source locations, most of our traps, they're going to have one connection. Those are our sink locations. And we identified our source location as the CC-21-27695. That's going to be a non-routine trap within the Maricopa County trap network. So this is a trap that was set out following a resident calling and reporting a bunch of mosquitoes in the area. And this was actually located within a desert neighborhood in the far north portion of the county. And so it's not an urban landscape, it's going to be a more desert landscape. And then also RT1017, the green trap, that's going to be our trap that has a lot of connections. It's also contributing a lot to the network. And this is actually down in the city in a more suburban area next to some man-made lakes. And now let's look at population 2. So population 2 is going to be an eastern or southeastern part of the county population down in like the Chandler, Gilbert, Queen Creek area. And when we perform our network analysis on this population, like our last analysis, most of our traps are dead-end sites. They're just our sink sites, not contributing to the network, whereas we have a couple that are contributing to the network. And so our source location is identified as RT207. This is going to be a trap site that is located in a suburban neighborhood in the city. However, that's not the site that's contributing the most to the network. So this trap location in the red, RT1020, that's going to be the trap site that is contributing the most to the network. And this trap site significantly is next to some farmland. And so this trap is, this farmland is probably allowing or providing perfect habitat for not only mosquitoes, because there might be more water there than any other part of the valley, but also it's providing good habitat for the birds. So providing perfect environment for West Nile virus to proliferate from. And going to population 3. So population 3 is actually our smallest population in 2021. However, it did progress into 2023. And this is going to be a population that was more in the western part of the county. And so when we look at the network analysis, in just 2021, we can see a majority of our trap sites are sink sites, and our identified source location is going to be right next to a wetlands location. And so yet again, this is probably providing ideal habitat for mosquito and bird, so great West Nile virus proliferation from this site. But we wanted to get more information from this population in particular because because we saw this population persist into 2023. And so we did a multi-year analysis where we identified most of our traps were sink sites, but we identified the exact same trap location in 2021 as our source. And we saw that connect directly into the same trap location in 2023. And so with this multi-year analysis, we are seeing that this population is persisting at this particular location year to year. And lastly, going into our fourth population, this population is going to be a largely it's kind of spread throughout the county. However, it's more focused in the northern part of the county. And we did see some introductions into Riverside County, California in this population. And when we look at this trap network, we can see yet again, most of our traps are sink sites, but our trap right there in the middle in the blue, that's going to be our source location. And it is also the trap that is maintaining the network and maintaining this population. And this trap was located within a suburban environment. And so for some conclusions for West Nile virus, West Nile virus is endemic to Maricopa County, overwinters and reemerges annually with a limited number of new introductions into the county. And West Nile virus from Maricopa County has repeatedly spilled over into other counties in the Southwest, as we saw with Clark County, Nevada, and Riverside County, California, making West Nile virus in Maricopa County not only a public health threat to Maricopa County residents, but also to residents in the Southwest. And then we identified most traps in our endemic populations from 2021 are not contributing to the maintenance of West Nile virus. So this can help us better inform our public health partners of the source locations that we identified for those 4 endemic populations. And one of those populations was maintained through 2023, and our source location appears to be maintaining our population year to year. And so now we're going to shift into mitochondrial phylogenomics of Culex quinquefasciatus, the vector for West Nile virus within Maricopa County. And so our goal of performing phylogenomic analysis is to determine the local and regional relationships, range, and potential population barriers of Culex quinquefasciatus in the Southwest over space and time through the utilization of pooled samples, so that we can collaborate with our vector control partners. And so we expected to gain a better understanding of West Nile virus maintenance and circulation within the region. So how are mosquitoes contributing to the circulation of West Nile virus, and how are birds contributing to the circulation of West Nile virus? So comparing the two. And then hopefully we can inform targeted mosquito control efforts. And so first off, how do our methods compare to current methodology for population genomics? And so current methods have relied on single individual sampling, so looking at one individual mosquito at a time. And they typically use microsatellites, mitochondrial genes. In a handful of cases, they've done whole genome analyses. And sequencing relies on short read sequencers such as Illumina or Sanger. However, we wanted to work with our vector control partners and work right into their workflows. And so we propose to use pooled sampling. However, we can't use short read sequencers in a pooled sampling scenario. Because of reassembling, we lose our resolution and we're not able to get individual allele frequencies. And so we decided to come up with a new target, and we are now amplifying complete mitochondrial genomes using a long-range PCR approach. And so we're getting, with one PCR, the full 15,500 base pair genome. And then we sequence that on a long-range sequence or a long-read sequencer. And we chose Oxford Nanopore MinION. And then because of the high error rate of the MinION, we decided to do error correction with Illumina sequencing because it has a lot higher accuracy and is the gold standard for sequencing. And so when we applied this methodology to a population of mosquitoes in 2020, so we took a subsample of our 2020 sampling and then took 48 of those subsample, pooled them together, And so that made a larger pool of 176 mosquitoes. We were able to identify 825 variable sites. And so if we look down on this plot, we have our mitogenome position in base pairs from on our x-axis from 0 to 15,500 base pairs, and then our read counts on our y-axis. And we have our max allele frequency in the black and our minimum allele frequency in the gray. Our minimum allele frequency is telling us exactly which positions those variable sites are present. I do want to point out where we have our large peaks in the 16S on the front and the back of this plot. Those are due to our primary binding sites, and we're seeing a lot higher coverage at those. And so if we were to edit off those primary binding sites and make this plot again, we'd see our average coverage go down in those regions. However, we wanted to look at what would this look like, or our methods look like in an ideal scenario where we're able to control or make sure that each individual within a pool is equally represented. And so we took 5 individuals and we quantified those. And then we constructed pools by pooling those individuals in different ratios or with different individuals into a pool in the same concentration. And so we have pool 1 with individuals 1 and 2, pool 2 with individuals 1 through 3, pool 3 with individuals 1 through 4, and pool 4 with individuals 1 through 5. We then amplify these individuals with our long-range PCR approach and sequence on both the Minion and Illumina. And so this is where, and then we looked at the number of variable sites and uncalled gap sites within both our Illumina data and our Minion data in both individual scenario and a pooled scenario. And we identified that within our individual mosquitoes, we saw a 480% increase in the number of variable sites called between Illumina and Minion. And we saw a 48.7% increase in our pooled samples from Illumina to Minion. And this is probably due to the higher error rate of ONT sequencing. It is around 10%. And so we're probably getting a lot higher number of variable sites because of the error. And so we're going to more trust the variable sites that were called in the Illumina data. And when we're looking at the Illumina data, we can see that when we move from individual to pools, we do see an increase in those number of variable sites as expected, because in the pooled scenario, we do have more diversity within that library. However, within our individual, we still saw 131 variable sites. This is probably due to heteroplasmy. And so one individual having more than one mitochondrial genome. However, Illumina data is not the answer, as we would lose our individual level resolution. And so moving forward, so we have shown that we can amplify the complete mitochondrial genome with both individual mosquitoes and pooled mosquitoes. And then we've also shown in our controlled pooling experiment that the Minion sequencing data on our pools is much higher error and we obtain more variable sites. However, the Illumina data with short reads leads to ambiguities when we reassemble. So we are moving to a PacBio hi-fi sequencing platform where we can sequence long reads up to 25 kilobases in length or 25,000 base pairs with the high accuracy of Illumina sequencing. And so we've actually sequenced our controlled pooling experiment on this platform, and we're currently working through our analysis. And with that, I can take questions.
Thanks, Zach, for that presentation. Looks like we have time for about 1 or 2 questions. If folks want to raise their hands or just go out and ask questions. So maybe, oh, there we go. Okay, so let's start with, so Chris, he's in the top left-hand corner of my screen. So let's start with Chris.
Hi, Zach. That was a really nice presentation. And one of the intriguing possibilities with some of the viral network elements like you were showing, and the fact that maybe Maricopa could be a focus that results in transmission in the broader region would be, if there's some relationship with the viral intensity or the viral population size in Maricopa if we could control that, then maybe that would preempt viral spread to other areas. And I wondered if in your analysis you can see any link between intensity of virus activity in Maricopa and when those leaps happen to other areas like Las Vegas, for example.
Yeah, so we would need to perform the network analysis on, I think, the more broad Maricopa County clade. And so we could kind of see when those jumps happened and what traps were contributing the most to that jump. And so I think we'd have to do a more broad scale analysis than what we did, because we just performed that analysis focused in on 2021 populations.
I'll just chime in with that as well and say that I think that doing affected population size over time to see if viral load is increased in Maricopa County, and then just looking at the dates where you start seeing positives pop up in the other counties, that would be a really good, really easy way to do that. Okay, Sylvie.
Hi, thanks for that talk. That was really interesting. I was wondering about the actual traps that you identified as being the most, like, the sources. And of course, you've been trying to figure out, okay, well, what is unique about those particular locations, right? So I was wondering whether what you think at least would be happening there, and particularly, is it the local location, or could it be that it's actually the bird population? And maybe if you do this over years, you're going to find that source moving around all the time. So the source might be a little bit more random and it's dependent on the bird population, or whether there's something like ecologically unique in those locations that leads to those sources.
Yeah, and I think that's a good point. So I think at those source locations, we could definitely have some maintenance. So like at the wetlands and at the farm locations, those are going to provide ideal habitat for not only our vector, but also for our reservoir hosts. And so those could be what's maintaining that population. But like you said, there could be birds moving that could be moving our source location around. However, that's kind of where we are using our mitochondrial population genomics of the vector to see, like, is the movement of West Nile virus more correlated to the mosquito or the birds? And so if we have high gene flow within that population, we would suspect that there is more of a mosquito influence within the maintenance of West Nile virus. And so that could be more of a trap location specific, since they don't have as large of a flight range as birds. And so I think looking at all of the scenarios, so like what the exact trap locations are there a lot of standing water at those locations, or kind of looking at the bird populations as well necessary.
Thanks, Zach. Jennifer Henke has the last question, and then we're going to move on to the second talk.
Hi, Zach. That was a really great presentation, and obviously a complex issue. I was wondering if it's a bit related to Sylvie's question. I was hoping that you could spend a little bit of time thinking through, why do you think you only had one area that you were able to see a year-on-year maintenance of that population? Do you think that perhaps we as vector control need to move our traps into different locations, or is this just that big of a complex issue that it's really hard to find that maintenance of what's going on from year to year in your virus isolates?
Yeah, and so this is something that, like, I thought about after performing these analyses. And I think potentially, like, it could be that one location is really good spot for our vector, our reservoir host, and it's just really good at maintaining West Nile virus. But also vector control, like, during sampling, there's a chance that we potentially missed those West Nile virus positives in those mosquitoes at that particular site within a sampling period. And so there could be we missed the sampling, so that population could be persisting. Or we're currently still sampling and sequencing 2023. So we still maybe haven't caught it as well.
One other thing, Jennifer, is that for this analysis, there was, we didn't include any clade that also had interactions with the other counties. And so those clades actually did persist into 2023, but we didn't want to sort of mess up this idea of a population by adding in those. So it's still in its infancy. Okay.
Thank you
All right.
for expanding on that.
Thank you, Zach, and thank you everybody for all the really great questions. Now I'm super excited to move on to the second talk. Again, it's training STEM students in mosquito surveillance and identification. And just to go a little bit further into the two students who are going to be presenting. So Abdoulaye Zerbo is a graduate student pursuing a Master of Public Health at California State University in San Bernardino. He got his bachelor's degree in 2021 and plans to deepen his expertise through grad studies to better serve the community, which is awesome. He's developed a strong interest in vectors, their biology, and their behavior. And then Mayra Espinoza is an undergrad, also at the same university, pursuing a Bachelor of Science in Environmental Health Science. And her research is also primarily focused on the vectors of diseases and the impacts of agrochemicals on their survival, development, and fitness. And just to reiterate, I think that it would just be so good to be able to have this training that I think that they're about to talk about in other places like Arizona, where we lack a lot of that expertise. So let's see, are you, Abdoulaye and Mayra, are you able to share your screen?
Yes.
Okay.
Hi, everyone. Are you able to see the screen?
We are. It's currently in sort of the normal PowerPoint, but if you wanted to, I don't know if you want to move it into a presentation.
Yeah.
Version. All right, looks great. Thank you.
Good afternoon, guys. My name is Mayra, and you guys just heard from my colleague Aziz. And today we're going to be presenting the Curricular Enhancement in Public Health Entomology, where we carried out a training for STEM students in mosquito surveillance and identification. The project was originally proposed by Dr. Nick, who's had many years of experience in the field of entomology, and it primarily focused on public health entomology, which allows us to gain a better understanding of the role that insects have on disease transmission to humans, which is crucial for the implementation of disease prevention and control strategies, which ultimately reduces the transmission of vector-borne diseases. In collaboration with Dr. Nick, which is the PI of the training, we devised a plan for structuring and executing a training program which focuses on mosquito surveillance and identification. The primary objectives of this training were to encourage students to enroll in Cal State University's Environmental Health Sciences program, as well as to prepare undergraduate students to work as mosquito surveillance technicians for local control agencies or organizations, as well as to encourage students to pursue, as well as to encourage students to pursue graduate studies in the field of public health entomology and hopefully cultivate new generation of medical entomologists and vector biologists. Mosquitoes significantly impact public health. Not only are they considered a nuisance, but they can also harbor and transmit the microbes that cause disease, thereby potentially adversely impacting public health by increasing the morbidity and mortality rates across the globe. So gathering critical information about disease vectors as well as early identification and monitoring are crucial for the implementation of timely control measures. Surveillance and identification allows us to assess whether there are mosquito-borne diseases in the area, their population size and dynamics, as well as the effectiveness of the control measures that were implemented, which enables us to create a more targeted interventions as well as implementing prevention strategies. In collaboration with Coachella Valley Mosquito and Vector Control District, the training program was conducted, and it was 3 weeks long from July 24th to August 10th, which focused on mosquito surveillance and identification. The program itself consisted of morning lectures and afternoon lab sessions from Monday through Thursday. It took place at Cal State University, San Bernardino, where students had hands-on training at the university's mosquito rearing facility. Students also had the opportunity to visit Coachella Valley Mosquito and Vector Control District in Indio on their final day of instruction, which was a good opportunity for students to see a real-world application of what they've learned during the training. The PI has a really strong background in research and teaching with a focus on vector-borne diseases and is actively involved in environmental health sciences curriculum at Cal State University, San Bernardino,
And along with him, the trainees played a vital role in shaping the training curriculum by creating materials and assessments necessary for the training, as well as assisting students in understanding the life cycle and development of mosquitoes. So we had a total of 10 participants from Cal State University, San Bernardino, and local community colleges, which came from diverse backgrounds in environmental health sciences and biological sciences fields, which made it a well-rounded and inclusive training program. Because our proposal was approved a month prior to the training, we had a limited amount of time to gather all the necessary materials and create an enticing flyer that would draw in students. We actually created multiple versions of the flyer before the final version was completed, and just a week before the training, we began distributing it. To advertise the training effectively, collaborators reached out to students and colleagues via email, and we also physically distributed the flyers, and we posted them around the campuses and nearby community colleges. The information on the flyer contained the training dates and times, which are posted here. And we also had the funding partner, learning objectives, collaborating agency, and a QR code, which made it easier for the application process. They would open up their phones and they could directly submit their application, which had their resume and their contact information, along with an area where students could show their interest in the training course. And then the trainees promptly responded to any potential participants who needed any clarification questions via email.
Thank you very much, Mayra. So on the left side here, we have a weekly breakdown of the training. It was well structured and clear, we think. And again, our main objective here was to make sure each topic receives the adequate attention, which would contribute to the overall success of the training. We also thought it was very important to include the map of the campus, to show where the building, where the training will be held at, and also the closest parking lot so they don't get lost in the process. Materials. So in the materials, we had a welcome message from the PI. We also thought it would be very useful to create a shared Google Drive because we wanted all the participants, again, to have access to the materials at their own convenience, using the technology, of course. They were able to access the lectures on various topics. Also, we included the Key to the Mosquito Identification of California, 2021. Within the materials, we also had a post-training survey. So as every training, we wanted to gather feedback because it helps assess the effectiveness of the training and also identify areas where we needed to improve for future training. We've also included in the materials the scheduling for the Coachella Valley, the tour to the Coachella Valley and mosquito vector control district in India. Assessment. So we had a pre and post assessment, each with 20 multiple choice questions on various topics, mosquito biology, ecology, behavior, surveillance, identification, and control. So prior to the training, we needed to make sure we had all the different stages of the life cycle of the mosquitoes, right? So we made sure we had rafts. We had all 4 larval stages. We had pupation. We also had adult mosquitoes because we wanted them to have a visual and everything. Let's go with the weekly breakdown. So we started the training with a pre-assessment. Pre-assessment because we wanted to have an idea on the current knowledge on these topics, right? And perhaps set the stage for more personalized and effective learning experience. We also had lectures on various topics, such as behavior, mosquito behavior, host seeking, vectorial capacity, and all that. And the week one also, in week one also, they were able to visit the rearing facility located at the CSUSB, where they were able to see the different materials that we use and also familiarize themselves with the different stages. And what we did, we divided them in 3 groups, and we had each group collect first instar because we wanted them to see the evolution all the way to adults, have the full experience. In week 2, they've continued their hands-on project in the lab, and we also had a high-quality surveillance and control training delivered by Jennifer Henke. She is the laboratory manager at the Coachella Valley Mosquito and Vector Control District. She provided a very informative training on surveillance method and some equipment assessment. Students also were able to take some traps home to see if they have any hot spots in their homes. When they came the next day, we were kind of laughing because some people even set the traps in their garage, and they were surprised that they collected so many eggs in it. We thought that was very interesting. Week 3, we've switched gears a little bit. The lectures were based on mosquitoes-borne diseases. They've learned a lot about malaria, dengue fever, Zika virus, Zika, and West Nile. They've also had some practical sessions on mosquito identification using the key to the mosquitoes identification of California. They've also had a guided tour to the Coachella Valley Mosquito Vector and Control District in India, where they were able to visit the facility, familiarize themselves with the different materials used over there, equipments. And when they came back from the tour, they took a post-training assessment because we wanted to see what impact the training had on them. So after completion, they receive a certificate of completion from the Department of the Health, Science, and Human Ecology at Cal State San Bernardino. Analysis. So on average, the students scored about 51.5% on the pre-assessment. Again, we wanted to have a broad idea on their knowledge. And the post-assessment showed increase, went to 77.2%, which testifies the impact the training had on them, right? One of the students even, one of the participants even decided to pursue her own research interest. She talked to the PI and, she was able to start her own research. Challenges. The big one is time. The grant approval just arrived a month prior to the training. So we're scrambling everywhere, trying to create the flyers and advertise. And it was quite challenging. Financial constraint also. The daily parking, because we didn't have the funds like on a timely manner, the trainees had to pay the parking permit out of their own pocket, which was a little bit of a burden for them. We understood that, but we were able to compensate that with some gift cards after the training, and even the refreshment and snacks were provided by the PI. Also, we had some transportation problems. So initially, the tour to the Coachella Valley was supposed to be on the 2nd week, but due to some logistic problems, we had to postpone that to the 3rd week because finding a van that could accommodate all 10 participants were a bit challenging. Conclusion. So this intensive training in mosquito surveillance and identification provided participants again, with a deep understanding of mosquitoes biology, their behavior, and control techniques alongside with hands-on experience. So we think this training was able to address some critical needs for well-trained individuals in mosquito surveillance control. Also foster career aspiration, because most of the trainees were undergrad. So why not pursue their graduate studies and start their own research? And we also think this training help, in public health entomology, and also will potentially help professional development in the field. Some of the recommendation, time is a big one. For future training, we'll probably need more time to reach a wider area of audience, which will increase participation, right? And also we'll want to expand our collaborative network, have different agencies, bring their different resources, different expertise, which would benefit the trainees. And also incorporating small research projects into the curriculum to deepen practical and practical experiences and problem-solving skills. We would like to finish with the acknowledgements. We would like to give a special thanks to the Pacific Southwest Center of Excellence in Vector-Borne Diseases. A special thanks to Jennifer Henke at the Coachella Valley. Special thanks also to Dr. Nick Mahmoud. Thank you also for the tour at the Coachella Valley Mosquito and Vector Control District. Special thanks to our University of California State San Bernardino. We would like to thank Tori Krum, who is the animal health technician. And also a special thanks to Carrie Hartstrom, who is the administrative support assistant for the Department of the Health Science and Human Ecology. And thank you very much. We will gladly take any questions if you have.
Hey, we'd love to have some questions. Oh, let's see. I actually didn't mean to raise my hand, but there we go. Jennifer, do you want to go first?
Oh, I was just trying to clap. Great job, Aziz and Mayra. You guys did an excellent job on sharing all the work, and I'm so glad to see the assessment that you did. Thank you for sharing it.
You're very welcome.
I do have one question that I'd really like to ask. I'm just wondering, do you feel like there was a part of the training that the students really gravitated to the most? And then also in the reverse, was there something that you feel like they were least interested, that you feel like you're going to have to work on a little bit to bring to get more interest from the students?
So I definitely feel that the students enjoyed doing trapping and surveillance a lot more, just because I feel like it was more in the field, hands-on training. So I feel like maybe applicating, like bringing or setting traps and then having them bring back the traps, and then identifying their own mosquitoes from their traps. I feel like that would be like a great growth for them, because just identifying the specimens that we did have there, it kind of wasn't as exciting as going out, setting your trap and then coming back, which is why I feel like maybe they didn't enjoy it as much. But definitely it would be like a good experience to add on to the training for future.
Oh, thank you. And Chris?
I'm curious how aware the students were generally of vector control and careers in vector-borne diseases when you started this. Do you feel like it raised awareness, or were they already aware of the options?
So some of the students that actually joined the training kind of had a little bit of knowledge within it, because they've been taking courses with the PI himself. So they kind of had a little bit of general knowledge, because he has a lot of love for insects and he kind of like tells us about it all the time. But there was some students there who I really don't know how they ended up there. I think it was just because they read the flyers or they heard of it somehow. So it was nice to see like more inclusivity, because we only really saw maybe 2 students who already previously had a background. And then the other 8 were just kind of like reeled in.
really great presentation. And I think programs like this are a big point of pride of our center and really ideally reaching broader audiences. And Jennifer and Coachella really also appreciate your contributions to that, because these are rich experiences that we have a hard time providing at the university always. I think that collaboration is really valuable.
It looks like we have another question that was put in the chat by Nicole Foley, asking, what was your most successful method of advertising?
I actually feel that kind of going out there and like talking to people and just putting up the flyers, like that gave everybody an opportunity. Because even if that person themselves couldn't join, like I would always tell them like, oh, make sure you tell your friends, like this is a really good opportunity. It's free. It's like, nobody's going to charge you anything. And the only thing you're really going to gain is growth and experience from this, which could, give you a job later on in the future.
That's awesome. Looks like Dr. Nick would like to make some comments.
Thank you very much. I just wanted to say that reaching out to STEM offices and a STEM department of every college was a good idea, because, in every community college, they have an office for STEM, and they have similar activities in different areas. And I, fortunately, because of a prior project, I was in touch with many of them, and I could very quickly, send our flyer and ask them to advertise it everywhere when they can. And that was the reason that we could bring some students from the community colleges around us. And that was the main goal, to make more students interested in vector-borne diseases, public health entomology, and, the field of medically important arthropods, and, in general. Because they are the future of these professions. If we can attract more people to this field and make them more interested, there is a higher chance that at least some of them in future join us and join this business and continue the work in future.
Great. Irene Roberto from ADHS also asked, can you share the training resources, or is that something that you're planning on making available in the future?
When we had the training, we, as is mentioned, we had a Google Drive that we put everything there, and then sure, definitely, we can share the lectures in PDF, for example, and the key, of course, is I think it's available in PDF to most people. They have it. Also, we had a couple of other internal like manuals or these things that we used in our classes, and these things that we improved it a little bit and polished it a little bit and adjusted for this training and shared it with the students. And the rest was just going on in the lab with mosquitoes, or bringing the mosquitoes to our teaching lab. So they learn how to work with the key, what a key is. That's the first thing, how to work with it. What are the taxonomic characters, how we have to find them, and then little by little go towards the key. So identification was a big portion of this. And for surveillance and control, we received a lot of support from Coachella Valley mosquito vector control districts, and again, a special thanks to Jennifer and Jeremy Whitty for their enormous help. Yes, definitely we can share the lectures and resources and those things that we had in PDF.
Great, thank you. Oh, it looks like Mayra did actually share the Google Drive, so that's exciting. Are there any last questions that anybody wants to ask? Okay. With that, I think I'll just say that this is a really great showing for our first fall training seminar. So thank you everybody for joining. Thank you to the speakers for giving us some great talks today, for kicking off the season, and thank you to all of the partners, PacVec partners, for joining the call. Chris, did you want to add anything?
No, that's great. Thank you for hosting.
Yeah, great. All right. See you, everybody.